Welcome everybody to Paleo Talks. This is episode 16. Really happy to have Dr. Julie Meachin with us today. Hey, Julie.
Hi.
And today we're going to be talking about caves and a natural trap cave actually in Wyoming. And before we get to that, introduce our science communicator, David Moscato. And David usually gives us a little bit of an overview of how the show works. If you could do that, David.
Sure thing. So anyone who's watched the show before will know, but as a reminder, what we're going to do is we're going to start off by introducing our guest and going through the presentation. And then about, 25, 30 minutes in, we're going to open it up for audience questions. So we'll make a reminder when that time gets close. And then if you're watching us on Facebook, you can leave your question in the comments. If for some reason you can't comment on Facebook, you can find us at Gray Fossil Site on Twitter or Instagram and send us your question there. I'll be keeping an eye on that. And then we'll get through as many questions as we can before we let our guest go so we don't take up too much of their time.
All right, thank you, David. So we're coming to you from Johnson City, Tennessee, East Tennessee State University, and the Center of Excellence in Paleontology. And as we mentioned, this is episode 16. So we continue to have these episodes out to you weekly on different paleontology topics. And today, some of my favorite topics, Pleistocene animals, cave sites, and Dr. Julie Meachin, who's going to talk with us. And Julie, if we could just start out by you telling us a little bit more about the position that you're in now, where you got your degrees.
Sure. So currently I'm an associate professor of anatomy at Des Moines University, which is osteopathic medical school. I've been here since 2013. And before that, I did my undergraduate degree and my master's degree in zoology at the University of Florida, where I did some work at the Florida Museum. And then I went on to UCLA to do my PhD with Blair Van Valkenburg on big carnivores. So I'm a big carnivore person, much like Blaine. And then after that, I did a couple of postdocs. My first postdoc was at the National Evolutionary Synthesis Center at Duke in Durham, North Carolina. And then I did a year at Marshall University as an instructor for medical students teaching gross anatomy there. So hence why I got my position at DMU.
And Julie, didn't I hear on Facebook that you're starting a graduate program or having graduate students or something like that?
We are. So we just got accredited by the Higher Learning Commission to start a master's degree program in paleontology and biological anthropology. So that's where the strengths in our department lie. And so that is what we're starting. We're actually starting this year. We have one student, but hopefully next year we'll really get our feet under us and we'll be able to accept a couple more students into the program. It's really exciting.
Yeah, that's always exciting to hear of a new program growing. All right, well, we could go ahead and get started with your first slide if you'll go ahead and share the screen.
Yeah, let me do that. Okay.
And then my first question for you, once you get that up, is, what led you down the path of working at Natural Trap Cave?
So that's a great question. So my adventures at Natural Trap Cave actually started in about 2011, I want to say. And I didn't actually go to Natural Trap Cave in 2011. They started then because I was a postdoc at the National Evolutionary Synthesis Center. And I was a member of a working group there that was there for a couple of days discussing topics in Pleistocene megafauna and evolution. And actually one of the co-organizers, Alan Cooper, who was at the University of Adelaide in Australia, knew about Natural Trap Cave. He had done some ancient DNA there before, and he got really excited about possibly going back to get DNA right from the source. And so he needed not only an American colleague, but preferably a morphologist, somebody that really was a paleontologist that could go in and sort of lead everything. So we discussed it and we came up with a plan to go back. And ever since then, we've been working there. I wrote a grant for the National Science Foundation to go there, and it finally got funded in 2014. So that is how I began my adventures into Natural Trap Cave, and you'll be able to see through my talk that it's sort of snowballed into something much bigger than just that ancient DNA piece.
Speaking as somebody who's been in there, I'm guessing part of the reason that ancient DNA is well preserved is because it's so freaking cold in there.
Yes, so that was the thought, right? It's kind of like a refrigerator. It's very humid and very cold. In the dead of the summer, when it's almost 100 degrees on the surface, it's only about 40 degrees in the cave. So it really is like rappelling into a refrigerator. And it is also really humid. So despite the fact that it's cold, there's a lot of water saturation in the air. And those conditions are sort of really good for preserving ancient DNA. They have this sort of mummification effect on a lot of the specimens. So the DNA is exceptionally well preserved. Although we have found that in the areas that are directly underneath the hole where they get a lot of precipitation, those specimens aren't as good quality as we would have hoped.
Well, if you could go ahead and start walking us through your presentation, I'll interrupt you every once in a while with a question.
Sure. All right, let's make sure I can get through. All right. So I just like to start out talking about why Natural Trap Cave is special. Why do we care about this site? One of the really interesting things about Natural Trap Cave is that we have a continuous fossil record from before the last glacial maximum, which is at least 30,000 years ago to the present. We have this continuous fossil record in this cave. We really have a lot of fluctuations in climate at this site and throughout the late Pleistocene. So this is just a curve of climate, basically taken from the Greenland ice core data. And this red box here is the time period that Natural Trap Cave encompasses. And you can see that there are huge fluctuations in climate during this time period. So it's really interesting for that reason. We thought that Natural Trap Cave captured the end Pleistocene extinction event, but actually it turns out it might not, which is really disappointing. But we're kind of hoping that we can find that end extinction event somewhere in the sediments. And it's really got good conditions for ancient DNA preservation, like we just talked about. It's constantly cold, less than about 42 degrees Fahrenheit, high humidity, very little to no human activity, and few other disturbances because it is really protected. And as we'll see in a bit, the site is actually gated off, and so nobody can get in unless you have a permit to get in and do that work. So I figured we could take you to Natural Trap Cave. Natural Trap Cave is in Wyoming, which is highlighted in black here on the map of the continuous US. Natural Trap Cave is in Bighorn County, which is right here at the top of the state in the middle. And then the site is actually at the very top of Bighorn County. So if there wasn't a giant canyon in the way, you could actually walk to Montana from the site. And if we are gonna go there, we might as well drive up the road. The road is actually really interesting. This is probably one of the nicer parts of the road. It takes a 4x4 to get up this road. We've had some mishaps with vehicles, a couple of pop tires, a truck that ran out of control. We've got all sorts of things that have happened on the road. But if you're driving down the road to the site, you'll see that it's sort of looks like a runway almost, like a clear path to the cave from this view. And here is the actual cave itself. So you can see, I want you to notice two things about the cave. First of all, you'll notice right away that there's this big old rusty grate over the cave. This was installed by the Bureau of Land Management in 1971. And that was in response to somebody almost driving their VW Beetle into the cave. So it actually had to get winched out. Nobody was hurt, thank goodness. But just to prevent any unfortunate accidents from occurring, and also to prevent people from going into the cave unauthorized, they installed this grate. The other thing I want you to notice here is that there's this little lip here. So this whole area is made out of limestone. The cave is a limestone cave. It's basically a giant sinkhole. And there's this lip of limestone here coming up to the cave. Now, if you were at this site in the Ice Age, when this grate wasn't there, you actually might not even see the hole from this vantage point. And that's really important for how the animals got into the cave. So basically you saw that sort of runway leading to the cave. What we think happened is that animals were running. Herbivores may have been running, carnivores may have been chasing them, and they're not paying attention to where they're going. They don't see this hole in the ground, and they go over this lip and then just into the hole. And so they go into the hole and then they fall about 85 feet, and they likely die, if not get injured irreparably. So just a little bit about the history of the cave. This fellow is Larry Martin, and he was a professor at the University of Kansas in the 70s, 80s, and 90s. And he was one of the first people to excavate the cave. So he and a professor from the University of Missouri named Miles Gilbert started excavations in Natural Trap Cave in 1972. So 72 or 73. And they went out there and they dug in Natural Trap Cave continuously every summer until about 1985. So they spent a lot of time out there and they excavated a lot of Pleistocene fossils. And all of the fossils that they found are now at the University of Kansas Museum. And this is just a picture of what that grate looked like in 1974, 1975 when they arrived. You can really see this lip up here. So you can see how big of a drop this is. And interestingly, so you can see the hole here where they went in. That's not the hole we used. I'll show you where our hole was. It's right here. They actually set up scaffolding. So they built like construction scaffolding so people could actually walk down the 85 feet into the cave. From what I understand from people who were there at the time, it was pretty rickety. It felt pretty unstable. I don't know if I would have personally enjoyed walking down scaffolding like that. So we don't use a scaffolding to get into the cave. We actually rappel in, hence my title. So this was our team in our inaugural year in 2014. Nobody has been in the cave before that since 1985, when Martin and Gilbert left the site. And you can see this is the door that we open up to get into the cave here. So we're all sitting here and we're kind of having an orientation from the BLM in this photo, and we have yet to go into the cave. So this is the cave. This is what it looks like when you open up that grate and you go inside. This is about 75 feet and we have about another 10 feet up at the top here. You can see actually somebody standing up here. This is a person. So that gives you a little bit of scale. It's probably our main caver, Juan Layden. So he's up there and you climb down this ladder. You're already geared up. You've got all your gear on. You climb down this ladder and you basically step out onto this little ledge that kind of slopes into the cave. So it's not really sturdy feeling, I have to say. And you turn around backwards after you're roped in, and then you step off the ledge. And the feeling that you get stepping off the ledge is actually stepping into the abyss, because it's so sunny at the top and it's not as sunny in the cave. So it basically appears black to your eyes. So once you actually step off this ledge and you're rappelling on this rope, you actually adjust to the light in the cave, and you realize that you're not in the abyss at all. You can see the floor. It's not nearly as scary.
One of the things I remember when I was there is just the high degree of safety, the way that it was set up, and the way Juan made sure everybody was taught and trained how to get in and out.
Yes, and that is really important, not only to the Bureau of Land Management who runs it, but also to the scientists who are doing the work. Mm-hmm. We all want to make sure that we're really safe. And I've also never had a sense of feeling unsafe while doing this work. So here's me on rope. This was definitely a picture from the first summer. And this is me rappelling in, and Right behind me here, there's a bunch of sticks. This actually is a giant packrat nest, and the packrat nests are really important to us because the packrats aggregate all these teeny little fossils, and they have for thousands of years. And they are basically the collectors of the microfaunal fossil record from natural trap. So who do we find there? We've got lots of different megafauna. So one of my favorite critters is this guy here with the red arrow. So this is Panthera atrox, the American lion. It's the largest cat, true cat, to have ever lived. Seems like they reached weights of up to about 450 kilos, which if you want to know what that is in pounds, it's roughly 900 pounds. So it's a 900-pound cat, probably a little bit smaller than that, but the artist who drew this is modeling here for you. And you can see that this cat standing on all fours would come up to his shoulder. He's probably about 6 feet tall, so I'm only 5'4". I'm guessing that that cat would be almost the same height as me. So that was a big cat. They did coexist with humans. We also have this guy. So this was drawn by Mauricio Anton, who just left ETSU. This is a picture of the American cheetah-like cat, Miracinonyx trumani, which is sort of thought to have a cheetah-like ecology. And here it is chasing an antelope caprid species. This may actually approximate what the landscape was like at Natural Trap.
Is it correct that some of the most complete Miracinonyx material comes from Natural Trap Cave?
Yes, absolutely. Natural Trap has the best fossil record of Miracinonyx.
It's the only place I've seen good mounts is at the museum at KU.
Yes, yeah, they have a full skeleton of Miracinonyx mounted. It's not all the same individual, it's sort of a composite, but it's all from Natural Trap. So we have several, basically we probably have several complete individuals amalgamated from Natural Trap, which is more than any other site that I'm aware of. We also have these guys, and no, they're not zebras, although they kind of are illustrated like zebras. These are native North American horses, and we have two genera of North American horses at Natural Trap. We have the genus Equus, which most people are going to be familiar with, which is your normal, it's your normal looking horse. And then we also have this interesting horse called Harrington Hippus, which is a stilt-legged horse. And those seem to actually be more common at Natural Trap than the Equus does. And they have just been started to be studied much more thoroughly. And we are also doing some more work on them for Natural Trap. So I'm excited about those. We have two different species of bison. We have this antique bison, which we find in Pleistocene sediments. And we have the modern living bison, which we find in Holocene sediments. So we do have sort of a chronospecies of bison at Natural Trap. We have these guys too. This is the Western camel, or the yesterday's camel, excuse me. So yesterday's camel is found at Natural Trap. And here's what it actually looks like in the pit. So the pit is the main dig site that was used in the 1970s and 80s by the teams from Kansas and Missouri. And we have not gone deeper in this pit. Instead, what we've done is actually gone laterally to this pit, and we find a lot of fossils. This whole area has now been excavated, and there's tons of fossils up here. So we have lots of different fossils laterally from this pit. And if you want to know how deep this pit is, this guy right here in the white hat is about 6'5". So this pit is probably 8 or 9 feet deep. And there's this ladder here, you might notice. We call this the ladder to nowhere. It actually was a ladder that went down into another chamber in the initial excavations, and just through time and sediment slump has been filled by infill. And now the ladder is basically stuck. It's not going anywhere. It's probably about a 6-foot ladder. And if you're wondering what the sediments look like, here they are. They're very fine, silty sediments. And all these things that you're looking at here are bones. So this is just basically me identifying bones that I can see, that I know what they are. And then all of these other things are also just bones, bone fragments. So it's a really rich sediment where bones are just everywhere. They're piled on top of each other. And unfortunately, we don't get a whole lot of articulated specimens. And the reason for that is not quite clear, and we're actually working on that as a question, why we don't get more articulated specimens. I mentioned before that the packrats are the collectors of all these little stuff. And here's the packrat up here. We still have those in the cave. They're still collecting things. They're still, you know, making fossils for future generations. And we have lots of rabbits. We have lots of rodents. We have small weasels. It's a cave, so of course we have some bats. We have lots of snakes and lizards, including the horny toad lizard is the lizard that we have in the highest abundance there. We also have birds. Birds actually fall in and die, believe it or not. We have lots of passerines, we have corvids, owls, raptors. And then weirdly enough, we actually have fish and frogs. And we think the reason why we have these, the nearest water source is probably about a mile away, at least. And we think the reason why we have fish and frogs is because different organisms will eat them, especially birds. They will release their droppings in the area, and then the packrats will actually go and collect all their droppings and bring them into their nest. So we think those fish and frog fossils are being collected by packrats. So like I mentioned before, the reason why we went there in the first place was to actually look at some ancient DNA at Natural Trap. This is one of the biggest parts of our project. And all of these papers were actually published before we went back to Natural Trap, but they include things like the bison, the American lion, and these horses that were at Natural Trap. These papers were all published on those things before we went, and we are doing much more work now. So the things that we're actually looking at Natural Trap include a lot of the carnivores. So right now we're in the process of sequencing the canids of Natural Trap, which I'm going to talk a little bit more about, the American cheetah, the American lion, and the short-faced bear. And these studies were originally led by Alan Cooper, but he is no longer working on the project. So now they're led by Dr. Kieran Mitchell, who was Alan's former postdoc at the University of Adelaide. So he is following up on all these projects, and we're getting them all published. And so the species I think that's probably nearest and dearest to my heart are the wolves. I really love the wolves at Natural Trap, and you'll see why I really like them in just a moment. I mean, I like dogs in general, but these ones are really cool. So the question started with what kind of wolves are present at Natural Trap? From the original work that Martin and Gilbert did, they actually said that there was both gray wolves, so modern gray wolves, and dire wolves at Natural Trap. And I wondered if this was actually true or not, because this is weird. These two wolves generally don't coexist in high densities at the same site. Usually there's more of one than the other, so I wanted to know who was there and in what abundance. So I actually undertook this study, and I looked at wolf jaws from Natural Trap Cave, published this study in 2016. And what we found here is that actually, so these are 3 different groups of wolves that we already knew. The blue circles up here are dire wolves from Rancho La Brea. The green squares are modern gray wolves from collections that I measured from the American Museum. And then these red triangles here are a special type of extinct, here's wolves right on cue, my wolves. These are special, a special type of wolf found in Alaska. They're called Beringian wolves, and they're related to gray wolves, but their morphotype is much more like the dire wolf. So they're really big prey specialists from the Ice Age. And the wolves from Natural Trap Cave actually fell in here with these Beringian wolves. So this one might be sort of overlapping with some dire wolves, but the rest of them really seem to really be pretty well into this Beringian morphospace. So who are they? What, you know, we wanted to know, are they these Beringian wolves? They could be. So we decided to take some ancient DNA from these wolves at Natural Trap and figure out who they were. So what we found from ancient DNA is that dire wolves are actually present in low abundance. We've gotten ancient DNA from dire wolves at Natural Trap, but they are not, they're not who's really there in high abundance. So who's really there? We've sequenced 8 canids from Natural Trap Cave and found that all the canids have a coyote mitochondrial signature, and they have a wolf nuclear signature. So this is kind of weird. What's going on? They probably went extinct at the end of the Pleistocene and left no descendants, or did they? So what in dog's name is going on at Natural Trap? We came up with 3 hypotheses. The first hypothesis is that all wolf-like canids at Natural Trap Cave are coyote-wolf hybrids with a coyote mom and a wolf dad. And how likely is this? These samples that we took span 20,000 years, and all of them went extinct. So I find this scenario fairly unlikely. Hypothesis 2 is that all wolf-like canids at Natural Trap Cave are descendants from an original hybridization event, which could be more likely, and then all could have still gone extinct from Natural Trap Cave. But hypothesis 3, from the work that we've done, actually seems like it might be the winner. This mitochondrial signature that we are seeing is not really coyote, but what we think is happening is that all modern coyotes arose from the group of wolves that the Natural Trap Cave wolves also came from. So basically, these Natural Trap Cave specimens carry the mitochondrial Eve of all modern coyotes. And as it turns out, red wolves might be the key to understanding what's going on. So this is something that we are working on right now with the ancient DNA of Natural Trap Cave wolves. And hopefully this will be submitted for publication by the end of the year.
Julie, would this be excluding the one that looks like dire wolf, or is it including?
No, so this one, these are all include, so they include the ones that look like Beringian wolves.
I see.
But we only have 1 verified dire wolf from Natural Trap Cave at this time.
And it's
So most of these specimens are ones that look like those Beringian wolves.
Thank you.
Yeah, thanks for asking to clarify that. So that's mostly what I've got. I just want to give a little plug. If you want to see more about Natural Trap Cave, you want to see us in the field, rappelling, Wyoming PBS came out to the site in 2017, I believe, and filmed, and we've got this great movie. It's about 30 minutes on Natural Trap Cave, so if you do want to watch it, here's that link. You can also just Google Natural Trap Cave Wyoming PBS, and it will come up. It'll be like the first hit on YouTube. And just a little sneak preview of another thing that I'm working on right now. This is a 57,000-year-old wolf puppy mummy from the Yukon in Canada. And we are working on this wolf pup right now, and we hope to submit a paper on her tomorrow or today to Science. So this wolf pup is really cool. She has all of her soft tissues present, including her intestines. And we actually know she's a girl because we can see her external genitalia. So she's been preserved for 57,000 years, and she's still looking pretty good. So plug for my next project. I just want to thank all the people who worked on this project with me. And I'll put up this photo of all the awesome people that have helped us excavate at Natural Trap. And I've got plenty more material for questions.
Perfect. That's a perfect amount of time. I do want to go ahead and ask one more, and that one is
Yeah,
about the Miracinonyx DNA and
yeah.
whether or not you're actually successfully getting some.
Yes, so we are successfully getting some. Turns out we're not the only ones doing that. Beth Shapiro's lab at UC Santa Cruz has also gotten some Miracinonyx DNA from the Yukon. And so we're sort of both kind of looking at this Miracinonyx DNA. It's possible we may combine forces, sort of do one giant project on it, although maybe not. The findings, they're supporting the findings from before, showing that Miracinonyx is a close relative of the puma, the living puma. But I think we're even going to have better DNA than that. We may even have some nuclear DNA this time. At least one of the teams might. So I think that's going to be, I think it's going to be really cool to see, you know, what the Miracinonyx DNA can show us.
Well, this is just a reminder now that we're wrapping things up, that go ahead and send in your questions. I see that some are already coming in. Is that correct, David?
That's right. I've got a list of a few that have already been asked. So as a reminder, anyone who's watching, you can, if you have questions, we're going to start taking them for Julie now. You can go ahead and put your question in the comments alongside this video. And again, if you can't comment on Facebook for some reason, I'm also going to be keeping an eye on the Gray Fossil site, Twitter and Instagram. So if you can send us a question there, go ahead. While we're waiting for people to send in their questions, I've got a few already. I'm going to start with this one from Carrie, who I believe is actually a question from Carrie's daughter, who has been here before. You mentioned earlier in the talk the humidity of the site, and the question is, why is humidity so important?
Well, humidity, so humidity is not actually that important for specific aspects, but one of the things humidity does is it actually helps preserve some of the moisture in the bone. Since it's so cold, the humidity actually sort of works in conjunction with that for preservational purposes. We don't want too much humidity. For example, we don't want bones to be waterlogged. We found that that really destroys DNA. But just sort of keeping those bones at that perfect temperature and humidity kind of mummifies those specimens quite a bit. So it's sort of kind of similar to the thought of like the bog people or those peat bogs or something like that, where those specimens are mummified. It's not the exact same process, obviously, but that sort of, they work together in conjunction.
I'll comment on that too for just a second. And one of the aspects with caves like this is it's the consistency. And when you don't have sort of dry, wet, dry, wet, dry, wet occurring, either if it's dry or if it's cold, if it's consistent, you're going to have better preservation.
Thanks, Blaine. Yeah, that's a good explanation.
David, why don't you ask another one there?
Sure thing. Donna asked, can you tell from the sediments in the cave when the sinkhole opening occurred?
So that's a great question. We don't actually know the answer for sure. We can make a good guess based on the fossils that we find inside. So we know that the limestone cave formed a very long time ago in the Mesozoic period. So basically, well before there were a lot of mammals that were roaming the earth. But the process of hollowing out of the cave, sort of that sinkhole process, took a very long time. And it could have had a crust on the top for a long time, even after that big hole formed. So we don't know exactly when that crust opened. But the sediments that we've got inside the cave, the oldest sediments that we have right now that we know of are close to 100,000 years. And so we know that the cave has been trapping things for at least that long. It could be even older than that, but we haven't discovered anything to let us know that it's older than that.
We have a question from Barbara asking whether or not any of the animals may have survived, if there's any evidence that maybe some of the large ones actually survived the fall.
So there is some evidence that animals survived the fall. There's a couple pieces of evidence actually. So we don't know the exact size where the animal would be irrevocably destroyed, right? It would sort of splatter. I'm imagining some of the bigger animals did, like bison and mammoths, probably didn't survive at all. However, it's possible that some of the carnivores actually did survive the fall, probably not without serious injury, but there's some evidence that some of the animals actually fell into the cave, and the cave sort of slopes down like this over about a football field of length, maybe a little less. And there have been fossils found at the very bottom of that deposit, which no animal would just fall into on their own. They would actually have to walk there, or sort of, you know, kind of creep along down there. So it's possible that there were animals that fell in and sort of stayed alive and kind of trudged their way down to the bottom there before they died. And then the other evidence we have is we've actually had animals fall in while we've been in there. And the smaller ones do survive. So we've had a snake and a deer mouse and a wood rat or a pack rat fall in while we were in the cave. The pack rat did not survive. The pack rat died. The deer mouse lived, and so did the snake. So they're both pretty small. So small animals certainly could have survived the fall, and large animals could have too, but probably not without serious injury that would have eventually caused their death.
Other than rodent gnawing, is there any evidence of any of the bones being chewed on?
So that's a great question. There was some talk about that from the folks at Kansas. So I spoke with people at Kansas, and this was all anecdotal, that there were some carnivore gnaw marks on some of the bones that they had originally excavated at the University of Kansas. I have not actually seen these bones, and we have not excavated anything that looks like that. So, like I said, it's all anecdotal. It's not, there's no hard evidence of that. But they swear that there were some carnivore gnaw marks on bones. So carnivores got in and survived and sort of gnawed things.
We have a couple of questions about the dating on the site. So Melissa and Grant have both asked about how the dates of the site are determined and if you know the sedimentation rate.
I do not know the sedimentation rate. Turns out I'm actually not a geologist. We are actually working on the sedimentation question. So we do have some folks, Dave Lovelace from the University of Wisconsin is heading up that part of the project, looking at sedimentation, along with my former postdoc, Corey Redman. So they're looking into that. For the dating of the specimens and the cave, we've got all sorts of different kinds of dates. We've got direct radiocarbon dates on the fauna themselves, and those range in age from about 41,000 to well into the Holocene, so 2,000 or 3,000 years. And then we also have some volcanic ash dates. One of those is about that 100,000-year-old date. That's one of the volcanic ash dates. And we have some OSL dates, which is optically stimulated luminescence. So it's basically looking at the last time that light hit sediment particles in the cave. And those dates jive well with our radiocarbon dates, actually. We took them from the same sediments that the fauna came out of. So those are kind of the date ranges we have and how we date everything in the cave.
Julie, we have a couple of people who are asking about the potential of volunteering and future field work.
So we will be having 2 more field seasons at Natural Trap Cave. I have funding for 2 more seasons. We had to cancel this year's field season, obviously, due to COVID concerns. It would be awful to get 50 people out there in the middle of nowhere and then have somebody come down with COVID. I think that would be a disaster. So we postponed the field season this year. So hopefully we will have a field season in 2021 and 2022. We do take volunteers. However, most of the volunteers we accept at this point are people who have already been out to the site and have committed themselves to doing that work. The only exceptions being new students that we take out there. And then we do have a couple of arrangements with a couple of museums in Wyoming to take groups of volunteers out there. And those folks maybe have never been out. But we generally don't take a lot of volunteers that we aren't familiar with just because of the nature of the site. So we will have field work and we do take volunteers, but they have to be vetted by us first.
Here's a question that I know Blaine will appreciate. And actually this question comes from Aaron Woodruff, who I believe has been out there
Yes.
to Natural Trap.
He has.
Aaron asks, any ancient DNA work on the short-faced bear?
Yes, so Blaine, I think you're working on this too. So I'll let Blaine take that question. Here's Aaron, right, for everybody watching in the audience, there's a picture of Aaron right here.
That's right. So Aaron and I went up there several years ago, and then I think he went back at least one more time as well.
I think he's been back twice more.
Okay.
I feel like Aaron's been there 3 years, but I could be mistaken.
Yeah, it's been a wonderful experience, I know, for him. So yes, Natural Trap Cave is definitely getting some ancient DNA from the short-faced bear that is there, but this is part of a larger project as well. Again, with Kieran Mitchell in Australia. And so that's a paper that we're working on with him and his postdoc, and it'll have some fun results coming out in the future. We also have some good material. Actually, the most southeastern material that we're getting is from Saltville, Virginia. So.
Awesome.
We've got a question from Mark, who's asking, have you found any carrion birds at the site?
Any carrion birds, so things like vultures is what I'm assuming.
Mm-hmm.
We actually haven't. We haven't found, as far as I know, I mean, it's possible that we have a few bones from carrion birds, but they are really not the majority of birds that we find in there. And I think the reason for that is that birds, I think, are loath to fly into the cave and fly into the bottom of the cave. We did find birds, little birds that can get through the grates. They actually nest right underneath the lip of the cave. So they fly in and out, but they never flew, they rarely ever flew around the cave. So they would fly out. I think that, you know, big birds may have been just not interested in flying in there for some reason. So we just don't find a lot of their bones. Sorry, my dog just freaked out over something. I have no idea. It just happened.
I know that you have some other slides, and one of the questions that many of us love to think about is the pronghorn and the American cheetah. And so if you could talk a little bit more about that idea. Again, I know you have some other slides that sort of cover that.
Sure. So this is a project that was led by one of my collaborators who's been out there with me pretty much every single year, Dr. Penny Higgins. She's an isotope geochemist, and she was really interested in the pronghorn American cheetah coevolution question. So this has basically been one of those just so stories in paleontology that a lot of people have told as to why the pronghorn antelope is the second fastest land mammal in the world. So the question she asked is, pronghorn, pronghorn, why dost thou run so swift? Here's basically a little overview of how fast things run. We have the African cheetah at 70 miles per hour. Here's the pronghorn at 60. And then we have a bunch of other fast species. But if you just look at North America, basically we have none of those species. So why on earth is the pronghorn so fast? And one of the reasons that people have postulated is that they evolved with this guy, the American cheetah-like cat. And that this cheetah-like cat has a cheetah-like ecology and runs very fast. And so this is why the pronghorn's fast, to get away from the cheetahs. Well, up until now, we've actually really had no evidence that this is true. It's really been a just so story. So these are all the just so stories. So what we did was we actually took stable isotopes from pronghorns, a couple other herbivores, the American cheetah-like cat, and the wolves. And we examined their isotopes to see who's eating who. And as it turns out, So here is basically a table with carbon and nitrogen isotopes. And these values in yellow are the ones that overlap. So here's the wolf that we have at Natural Trap. It overlaps with the pronghorn in nitrogen, but not in carbon. So likely wasn't eating those. It really overlaps with horses and bighorn sheep in carbon and nitrogen. So we think maybe it's eating these guys. But here's Miracinonyx, the American cheetah-like cat. It overlaps in both carbon and nitrogen with pronghorns and horses. So it looks like the American cheetah-like cat really was eating pronghorns at Natural Trap Cave, also eating horses. But this is basically the first evidence ever to show that Miracinonyx is actually eating pronghorn antelope, and not just a just so story. So we're hopefully collecting a little bit more data on this, and this is something that we hope to publish sometime in the near future.
That's exciting. Thanks.
Yeah.
Very cool. We have so a few different people are asking about specific types of animals from the deposit. So I'm going to throw just a few animals at you.
Yeah.
Let's see. Donna, Dick, and Maggie have asked about bats, weasels, and mammoths.
Bats, weasels, and mammoths. So we found all of those things there. I'll start with mammoths first because they're the easy one. Mammoths actually are in really low abundance. Most of the mammoth fossils were found by the Martin and Gilbert expeditions in the 70s and 80s. And there were only a few pieces of mammoth found, so very few mammoth individuals, which makes sense. Mammoths don't run a lot like other animals do, and they really are careful about their footing. So it makes sense to me that they rarely fell in, maybe during the winter when there was an ice crust over the cave or something like that and they couldn't see it. That's kind of our thoughts about when mammoths fell in. So we do have them, they didn't fall in a lot. Weasels and bats, we've got plenty of those. We actually have quite a few weasels. I'm really excited about our weasel diversity. That's something that's definitely ripe for somebody to do some research on, are the weasels at Natural Trap. And if I have a master student, that's one of the things I'm thinking about assigning them, is the weasels there. And then bats, we do find bats. I actually don't know what species they are. They're probably the same genus of bat that's there today. And so we haven't done a lot of work with the bats either, but they're there.
Would the mammoth be the Columbian mammoth?
That is a great question. And I believe that is what Martin and Gilbert said was there. But since we found so very little of it, we haven't done any comparative work on it. So I actually haven't looked into whether it's woolly or Columbian that are there.
To follow up on that, Julie, is there anything that you would expect to find that you haven't found yet?
That's a great question. Yeah, I'm kind of surprised we don't find more small felids like bobcats. I think we actually just found, we found some bobcat material, but it's really, really rare, which kind of surprises me a little bit. It might sort of be like the carnivore trap phenomenon where the smaller individuals, the smaller animals aren't trapped as frequently. But that's who I would have expected to see more of in there. That's not there. Mm-hmm. Otherwise, I mean, we don't find things that are in tree-like habitats or habitat with trees, but that makes sense to me because we really don't have a lot of trees at Natural Trap. It's mostly an open, dry environment.
And no ground sloth?
No ground sloth, no Smilodon, no saber-toothed cats of any kind. It's just too open. They need more of a closed habitat for feeding and hunting.
We have a follow-up to the cheetah pronghorn discussion. Eric has asked, and when I say Eric, I mean Eric Scott of Root, asks, have any biomechanical studies been conducted to determine how fast Miracinonyx might have run, or whether the pronghorns back then were faster than living species?
That's a great question, Eric. I think Eric already knows the answer, but to give everybody else the answer, one of the things that we actually have a lot of problem with is determining the physiology of pronghorns in the past. And that's because most of the things that, most of the organ systems, that basically give us an idea of their physiology are soft tissues, so heart, lungs, brain, blood vessels, things like that. So we would have to find some sort of proxy to determine what the physiology of pronghorns and or their relatives were in the past, and of cheetahs as well. One of the things that we have thought of is maybe histology on bone tissue, if there's some way to look at that and basically compare pronghorn to, the nearest living relative is actually a giraffe. So compare it to something like that, and also do the same thing with the Miracinonyx to see if there's any indication that bone histology might give us some sort of physiology proxy. The other thing that people have used is some other folks have used different blood vessels, like going into the skull, carotid blood vessels, going around the brain in different ungulates to basically say something about their cooling systems and some of their physiological adaptations. And those are maybe some adaptations we can look at in fossils to see whether they have some increased mechanism for increased physiological activity or being faster in the fossil record.
And related to that question, Ryan asked a little earlier, do we know how fast the American lion was?
No. The short answer is no. So we actually, of course we have an idea of how fast the African lion was. Here it is here. African lion can run about 50 miles per hour. That's probably their top speed. So they probably aren't doing that very frequently. Lions are not really known to be chasey hunters. They really like to ambush their prey more, short bursts of speed to get to their prey. So American lion may actually have been more open habitat adapted than the African lion from what we're seeing at Natural Trap. So it's possible that they may have been faster, but nobody has looked into that at all, as far as I know.
Interesting. Greg has asked, and again, this is Greg McDonald, who warrants a last name, I think.
Yes.
Do you have any mountain lion there?
We have not found any mountain lion at all. Evidently, there are mountain lions there now, but we haven't found any specimens that would make us think it's mountain lion. We have the Miracinonyx, and they're different enough from the mountain lion that we can distinguish them. They're bigger, for sure. But no mountain lion that we know of.
Julie, another thing I remember is that some pollen studies were underway. You have any results on that?
Yeah, so we've got some pollen samples. Mark Clements and Tom Minkley at the University of Wyoming are the two scientists working on the pollen that we have there. And we have a mostly complete record now from Natural Trap. So this is also one of the things we're working on getting published, is this pollen record. We have lots of different types of pollen from Natural Trap Cave, but the ones that really are prevalent are highlighted here. So we have pine pollen, which is highlighted in blue. We have Artemisia, which is sagebrush, highlighted here in yellow. And then we have Poaceae, which are grasses, highlighted here in green. And from these three genera, or these three genera, we can basically determine sort of the cyclical climate cycles at Natural Trap Cave, and that's one of the things that we're really trying to do. And so these pollen records are really good at helping us really fine-tune what the climate was at Natural Trap Cave when. And one of our projects is basically to line these up with our dates, our radiometric dates and our OSL dates and our volcanic ash dates, to basically understand when this is happening. So right now, we've got an okay idea of when the baseline is. That 110,000 year ash is one of those baselines. But we want to line those up with those dates to get a really good idea of what the climate was when at Natural Trap.
Do you have any sense whether or not there are differences of the microfauna sort of in conjunction with climate change?
So that was one of the things that we're looking at too, and we're also looking at morphological change in the microfauna. So those projects are underway. The microfauna project is being led by Dr. Jenny McGuire at Georgia Tech and her students. And what it really looks like is that we have a huge change in microfaunal abundance at the Pleistocene-Holocene border. So in the Pleistocene, we have far less microfauna than in the Holocene. And we don't think that's because there's not microfauna in the Pleistocene. We think that's a soil issue. Basically, the soil pH is, in the Pleistocene, is dissolving the fossils of the little things, we think is what's going on. And so the record is not being preserved as well from the Pleistocene. We do have microfauna from the Pleistocene. They're just not in as high abundances. And we're sort of looking at that change through time, like who is there when. And I'll give you a spoiler. We have tons of bunnies. And the bunny DNA is actually being run right now as we speak. It's being collated and analyzed. So we'll figure out all the different bunnies that we have at Natural Trap.
Right. you got the devil.
It's a cave bunny.
All right, we're going to start wrapping it up there. David, do you have a last one you want to try and do?
Let's see. I have two more that I'd like to ask before we wrap it up.
Sure.
One is following up from, we were discussing the big carnivores. We've gotten a couple of questions that are basically asking, do we have a sense of the relative abundance of different large carnivores, or how they may have been competing with each other or avoiding each other?
We do. So clearly at Natural Trap, I think the carnivore we actually have the most of are the wolves. So we have tons of wolves at Natural Trap. We actually have quite a few Marathonic specimens, and we have a decent amount of American lion. The carnivores we have in very low abundance include the dire wolf and the short-faced bear. So sort of those carnivores are in much lower abundances than the ones I just mentioned. So we do have an idea of who's more abundant there.
Okay. And then one more question I'll take. This will be another one from Carrie and Carrie's daughter. What is the oldest fossil at Natural Trap Cave?
Good question. We don't actually know the real answer to that, but I can tell you that the oldest thing that we've radiocarbon dated is a canid tibia, a wolf, a Beringian wolf tibia from the cave. It's 41,000 years old.
All right. Well, we will wrap it up. Julie, is there anything else you want to add before we stop the recording?
Just thanks for having me. It's been fun.
Yeah, no, I've learned a lot. All right.
Thanks very much. Thanks to everybody for joining us, for asking questions. We'll be back next week.
And just as a reminder, anybody who has questions that are outstanding on there that we haven't answered yet, we'll try and answer those and have Julie help answer those as well. All right. Thanks again, Julie.
Thanks, guys.
Bye-bye.
Take care.